Hello everyone, welcome back to another class from the sexual reproduction in flowering plants. So welcome back to my channel. In this video we are going to discuss about the structure of the pollen grain or the microspores. Now let's begin with the structure. Once the microspore will get mature, that will be converted into the pollen grains. So what is the structure of the pollen grains? There are various kinds of flowers, and in that varieties of flowers there are varieties of pollen grains. There are so many variations in their shapes, in their outline structures or ornamentation, which we used to call. So in all of that structure, what is the basic structure? In most of the cases you can find spherical structure in case of pollen grains, and they are absolutely similar. They are very smaller, sorry, not similar, they are very smaller in size, almost from 25 to 50 micrometer in diameter. Their sizes can vary. Okay, now if we'll see the structure of the pollen grain, there we can find that two membranes or double membranes are there. Now in these two membranes, outside membrane is considered as the XII and the inside layer is considered as intile. So let me write down the names first. Outside layer is considered as XII and inside layer is considered as intile. So the XII is made up of one particular form of chemical substance that is called as sporopollenin. Okay, sporopollenin. Now sporopollenin is very beneficial for the pollen grain, because due to this sporopollenin, as they cannot be destroyed by any kind of temperature, any kind of enzyme, any kind of pH, by nothing they can be destroyed. That's why the pollen grains can be fossilized very easily and they can be used as a fossil. After so long years also they can remain same as it is due to having this sporopollenin in their XII. And as this is the outermost layer of the pollen grain, so this is providing the protection to the pollen grain. Now let us move ahead into the inner structure, that is inner membrane, which is considered as the intine. Now this intine is made up of cellulose and pectin. There is cellulose and pectin. Okay, there is cellulose and pectin. These two materials are found, these two chemical substances are found in the intine. As because the stigma, in case of when we will study about the female reproductive part, we will see that there are three parts in the female reproductive part, that is the pistil, stigma, style and ovary, and in few cases the style is made up of the chemical that is called as pectin. So the intine must be releasing the pectinase enzyme to dissolve all the pectin so that the pollen tubes can grow and do the fertilization event. So we'll talk about it much later. Now let us focus into the structure of this pollen grain. And in few cases, in few spaces of this pollen grain, can you see that I have done a very thin layer, only the intine is present in few cases, in few space. So these space are considered as germ pore. These are considered as germ pore. What is the function of this germ pore? During fertilization, through this germ pore, this intine will be bulging outside. This can bulge outside from here and they can form this kind of pollen tube during fertilization and when the pollen grain will start to germinate. Okay, at that time the intine will bulge outside and they will form the pollen tube, and during pollination, during fertilization, these pollen grains will be germinating and that pollen tube will be formed through this germ pore. Alright, so this is the function of the germ pore, which will allow the pollen tube to grow outside. This is the overall structure of the pollen grain. Now let us see that inside view, that is anatomical structure of the pollen grain. If we'll find, then we will find that there are two types of cells which have been made. Two types of cell. One cell is absolutely bigger and another cell is very smaller in size. Alright. Now the bigger cell is considered as vegetative cell and this smaller cell is considered as generative cell. Now the vegetative cell, as you can see that this is much bigger and this has a very dense amount of cytoplasm. It can restore all the kind of nutrients in it, and when the generative cell, inside the generative cell there is the nucleus which will start to divide, which will divide mitotically to give rise to 2 more nucleus. Then they can, the nutrition can be provided to those 2 nucleus from this vegetative cell. Okay, so this is the reason why inside the vegetative cell the cytoplasm is so dense, and why inside that vegetative cell, inside that cytoplasm there are those are very rich in nutrients. Okay, now in the generative cell, in most of the cases the pollen grains will be shed. In this phase we can say that pollen grains are mature enough to dehisce. So once the anther will show the dehiscence, the pollen grains will come outside in this 2-cell stage. But in few cases the pollen grains can show further cell division, that is nuclear division. Sorry, that is not cell division, that is nuclear division. So this generative cell nucleus can be divided mitotically, and that can give rise to this kind of nucleus, which are considered as the male nucleus. And these 2 nucleuses are very important. And what is the chromosome number inside this nucleus? N number of chromosome. Because from our previous video we have seen that what is how these N number of chromosomes are coming into the pollen grains or the microspores via microsporogenesis. Okay, so as these 2 are having N number of chromosomes, these 2 nucleus will take part into the fertilization event, and these will be degenerated accordingly. All right, so here we are, and here we are ending with the structure of the pollen grain. Now due to having this kind of XII and various kinds of structural differences, various variations in them, they can cause kind of asthma, bronchitis, this kind of things can be happened in case of human beings. And apart from that, few human beings are there who have allergies to the pollen grains. But along with that, pollen grains are not only harmful, they are very beneficial also, because as you can see that they can store a rich amount of nutrients in them. That's why they can be used as a source of a rich amount of nutrients in the form of tablet, in the form of gels. They can be orally taken by the public. Okay, so that is also good for the public health. And they can be stored in minus 196 degrees Celsius, that is in the liquid nitrogen. They can be stored for a longer period of time, so that whenever it is required to do the fertilization event, then we can bring that pollen grain out of that store and we can do the fertilization events accordingly. Okay, so here we are, and one more term is very important, that is viability. That is up to how much time these pollen grains will remain viable. That also differs from species to species. In case of rice, wheat, we can find that only within 30 minutes, just after releasing from the anther, just after dehiscence of the anther, they will remain viable only about 30 minutes. This can be seen in case of wheat, in case of rice, in these plants. But in few other plants, they can remain viable for almost 1 long hour or 1 to 2 days also. And in few cases, they can remain fertile or viable for 2 days, 3 days, like that. Okay. So this is the overall view of the microspores or the pollen grains, and here we are ending about the male reproductive part of the flowering plant. From our next video, we are going to discuss about the female reproductive part of the flowering plant. So stay tuned to my channel, and if you found this video helpful, then please do like, share and subscribe to my channel. Thank you.